Hello and welcome everyone to the Patient-Centric Sampling Interest Group webinar. We're still waiting to see if a few more are going to join. There's still numbers, so just give us a minute and then we'll get started. Okay, it seems to be stabilizing. So, hello and welcome. My name is Jenny Royal and I work for a patient engagement and research company called Medipace, and I am also a member of the Patient-Centric Sampling Interest Group board. The Patient-Centric Sampling Interest Group is a non-profit organisation built by a community of amazing people, all focused on having a more patient-centric approach to blood sampling and advancing the adoption of using tiny volumes, microsamples, instead of tubes and tubes of blood for clinical trials, healthcare, whether it's diagnosis, monitoring, well-being, or something else. Today, I have the pleasure in welcoming you to the third in an ongoing series of webinars, and I couldn't have the webinars without our sponsors and partners. So I would like right at the very start to thank all of these sponsors who have contributed towards bringing this event to you today. In terms of what will happen today, I will be handing over shortly to Liana, who's going to talk for about 30-40 minutes before we open to questions and answers. If you have any questions during the presentation, please just ask me on the private chat. If you hit the general chat by mistake, it's fine. It's just we're trying to limit the distraction for others while they're listening, that's all. At the end of the presentation, we're going to have a question and answer session. Hopefully we'll get through them all, but if we don't, we'll try and address them afterwards. So have at it. Please share your thoughts, ideas, questions, and opinions. We would love to hear from you. The final thing I need to say before we get going is that this event is recorded, and the recording will be made available via the evidence base and the Patient-Centric Sampling Interest Group webinar so that your colleagues and friends can also access it as well at the end. And with that done, I am very excited to introduce Liana Goodwin, who is an old colleague of mine working at the Digital Experimental Cancer Medicines team, the Digital ECMT, as a research practitioner. And this group is very close to my heart because I was lucky enough to be one of those who helped it get going in the early days. Liana and her teams they build bridges between multiple worlds. So whereas most of us only have one view on things, they're linking frontline medicine with research, with technology, with patients, and they bring everyone together as partners to innovate around early clinical trials. It's amazing stuff. So Liana, over to you. The floor is yours.
Thanks so much, Jenny, for that lovely introduction. So let me just see if my next slide can appear here. Can anybody flag up that they can see that? It looks like it's just appeared. Hoping you can all see my first slide. Anyway, I will introduce myself in the meantime while that's loading. So first of all, thanks so much to all of you for taking the time to come along to the webinar and listen to me speak today. My name is Liana Goodwin, as Jenny mentioned, and I'm a research practitioner and I work in the Digital Experimental Cancer Medicine team, and I'm based at the UK NHS Foundation Trust, which is a cancer specialist hospital in Manchester in the UK. And today I'm going to talk to you about our work on in-home AKI monitoring. So just a brief synopsis of what I'm going to talk through today. I'll start off by very briefly introducing my team, the Digital ECMT, and my role within the team. I'm then going to move on to give some background on the in-home trial, more on that later. So I'll outline the design and the progress of the trial. And then finally, I'm going to give some information on how we plan to engage with patients and members of the public in our research going forward. My slides don't seem to be loading very smoothly. Okay, great. I can see my next one now. So as I mentioned, to start off, I'll give a really brief introduction on the Digital Experimental Cancer Medicine Team, or Digital ECMT for short, as it is a bit of a mouthful. We're basically an academic group, and we specialize in clinical digital research. So we work across a number of different research themes, as you can see from the diagram on the right here. But what's really key to our overall mission as a team is bringing patients, carers, and families together with all the different specialists who are involved in clinical research and work within it, and use technology to innovate and design new cancer care pathways as a group together. So Digital ECMT sits within the Cancer Research UK Manchester Institute, which means that our work benefits from the close collaboration that's taking place between Cancer Research UK, the University of Manchester, and the Christie Hospital as well. So as you can see at the bottom right of the screen, the website, our team web address is displayed there, and we do regularly post updates about our work on there. So that's a really useful resource if any of you would like to get any further information on our team that I've not mentioned here. Just waiting for my slide to load. There we go. Great. So transforming the role of the patient is a really key part of our mission as a team in Digital ECMT, and it's really central to our strategy moving forward. So our aim is to give patients the opportunity to become co-researchers and to actively participate in their treatment by being really directly involved in the development of new care pathways which involve technology. So this means that patients can be empowered to become much more involved with managing their own care, and can work alongside us to help design what we call the lab in the home, and to ensure that ultimately, and most importantly, this works for the patient. So what's also key to our goal of digitally empowering patients is making sure that the design of the technology that we use is patient-centered. For example, when we're using an app in order to deliver information, patients being involved in the design of that and how that works is really important, obviously, for usability and for it to be feasible to take forward. So when we're looking to use technology to develop all these sort of new and novel processes, it's so important that these work for patients before anything else. So what's really key to us being able to achieve this is our technology clinical trials. So it's obviously helpful, first of all, to tell you a bit about what technology clinical trials are. So when we use the term technology, this covers a number of different things. So this includes medical devices, or it may be software, and also artificial intelligence methods as well. Our trials are looking to test these different technologies under the same clinical trial conditions that are used to test new drug treatments. The driver in these trials is to ultimately help with clinical decision making to benefit the patient. And we look at doing this in lots of different ways, such as trying to change the design, delivery, and interpretation of early clinical trials, to develop new care pathways, and, as I've touched on already, to help to change the role of the patient in research through engagement and education. So at this point as well, I think it's also important for us to acknowledge that there is a digital divide that exists in healthcare technology, and this can limit for lots of patients access to these modified and new care pathways. And it represents a really important additional barrier to participation that's particularly relevant to our work and that we need to keep in mind through every stage of the process. So this is something that I'll touch on again a little bit later on. So a little bit now on my role before I move on to talking about AKI monitoring. So although the role of research practitioner is something that's quite well established within clinical research in the UK, my role in working exclusively on our technology clinical trials in Digital ECMT is something that's a bit novel. So as my background prior to moving into this role is more administrative and not necessarily clinical, I've taken on lots of different training to improve my clinical skills. And I'm involved in the whole process of conducting the trials. So I work across the patient-facing aspects and the administrative side of clinical trials as well. And this sort of balance of both sides of clinical research and sitting across both those areas is what makes the research practitioner role quite unique. So looking at the whole process of a clinical trial, as you can see on the diagram here, I'm responsible for each step of our technology clinical trials from beginning to end. So I recruit the patients to our trials, and that means obviously liaising with them and with the teams looking after them to understand whether they might be interested in participating. I answer any questions they might have and provide lots of detailed information on the trials that I work on, and I also receive their consent to take part in the trials as well. So once patients have given me their consent to take part, and they're confirmed as eligible for the trial, I then lead on arranging and completing training and education sessions for patients. These sessions tend to be quite detailed, and they're essential to ensure that patients are comfortable with all the novel processes that are involved in a lot of our trials. So trial kits are something that I often give to patients, and I'll go into that in a bit more detail later on. All the different items in these trial kits are explained and talked through with patients. And with things like blood sampling, patients might not be familiar with this at all. So sometimes I will not just explain, but demonstrate blood sampling as well as part of the initial training. And then when patients start participation on trial, I'm their sort of key point of contact day to day. I help them resolve any issues they might have with participation, arrange all the different assessments and appointments that are required for the trials. And then I actually complete these assessments as well. So this is where my sort of clinical skills come in, whether it's collecting blood samples or other samples from patients or completing clinical observations. And as I mentioned, administrative tasks are also a significant part of my role. So I manage all the data that we collect as part of our technology clinical trials and maintain all the documentation that we need as well. And working sort of so directly with patients, as well as being present behind the scenes of our trials, helps me to sort of really quickly gain feedback on various aspects of the trials, things that might not be working, things that are really positive. And then I can use that to sort of feed into the process to improve things in the future. So it's a really privileged position to be in. I'm not sure if you can see my next slide. Would anyone be able to let me know if it's displaying?
Yeah, we can see it.
Great. Okay, I can't see it, but that's absolutely fine. It's been a little bit glitchy for me, but as long as you can all see it and hear me, that's great. So in Digital ECMT, we connected with kidney specialist doctors who have an interest in what they call nephro-oncology. So that essentially means the care of patients who are facing issues with their kidneys, maybe due to the effects of treatment for their cancer or directly from also having cancer itself. So at the Christie, we do have nurse specialists who focus on managing patient kidney function during their treatment for cancer. But the renal specialist teams, so the teams that are dedicated to looking after people's kidneys day in and day out, are based at a completely separate hospital to the Christie. So this was quite a novel thing to bring these two specialisms of cancer or oncology and nephrology or renal treatment together. Kidney disease is something that we know from existing research can be a real barrier to treatment for patients who are diagnosed with cancer. And we look to work together with our colleagues in nephro-oncology to better understand this problem and how we might start to address it. So existing research in this area clearly shows that kidney disease is a major global health problem and that patients who are having treatment for cancer are particularly at risk of a specific type of issue with their kidneys called acute kidney injury or AKI. And this is for multiple reasons. So it can be the toxicity of the treatments for cancer themselves, or it can be dehydration in patients that comes as a complication of where their disease is and their ability to eat and drink, and how treatment can interplay with that as well. So in terms of the treatment of cancer specifically, we have got lots of new and improved treatments which are resulting in greater survival rates, which is obviously really fantastic. But this has also meant that there's been an increase alongside that in the development of both chronic, so long-term, and acute or sudden onset kidney disease. And also for the majority of cancer clinical trials, people with kidney function that sits outside of a very specific standard range or level are not allowed to participate in these trials. So when a patient attends a hospital, any patient in any hospital for blood tests, kidney function is one of the standard things that is checked as part of the patient's overall health when these blood samples are analysed. And for the reasons I've already mentioned, this is really particularly important for patients who are undergoing treatment for cancer. So one of the ways that kidney function is checked is through looking at the levels of something called creatinine in a patient's blood sample. So creatinine is a waste product that comes from the digestion of protein in your food and is also created by the normal breakdown of muscle tissue in the body. Creatinine is removed from the blood through our kidneys, and everyone will have some level of creatinine in their blood, but too much can be a sign of a possible problem with your kidneys. So there are other ways that kidney function is measured as well, including looking at a patient's urine output, for example, or looking at other blood tests. But creatinine is one of the key measures that's used in the NHS at the moment to monitor patients' kidney function. And in England, the NHS uses something called an algorithm to decide whether or not a patient is experiencing an acute kidney injury or an AKI at that point in time. And that's essentially a sudden decrease in kidney function. That's how we would sort of class an AKI. So what this means in this context is that the algorithm is basically a set of rules that a computer uses to look at the level of creatinine in a patient's blood sample, which is reported by the hospital laboratory system when they analyse the samples, and decide from that whether this means if the patient currently has an AKI. So put very simply, and I have to put this very simply because I have colleagues in digital ECMT who are AI experts, but I am not one of those experts. So put very simply, the algorithm does this by looking back at previous creatinine results for that patient and uses them as a reference result to compare to the current result. And for monitoring kidney function, it's really important to detect trends over time, which is why the algorithm looks back at any previous creatinine readings to determine whether an acute kidney injury is present. The diagram on the right of the slide here basically shows you the different steps that the algorithm takes to make this decision. And if the algorithm detects an AKI in the level of creatinine, it'll then place it into stages from 1 to 3 based on its severity. So with stage 1 being the least severe stage of AKI through to stage 3, which would be much higher in severity. So looking beyond how kidney function is monitored at the moment, if we were to increase the amount of monitoring that takes place, we know that we could better detect changes in a patient's kidney function. And not only sort of increasing the data in terms of detecting changes over time being an advantage, getting in there earlier and detecting a problem much quicker could result in improved outcomes for patients as well. One potential way that has been identified in existing research of doing some more intensive monitoring of kidney function is asking patients to self-monitor outside of the normal hospital testing schedule. And there's a real need to determine whether it's acceptable for patients to do this monitoring, to look at their own kidney function at home, away from the hospital, and then whether that process actually translates into clinical benefit for the patient, which is obviously ultimately what we're all looking to achieve. So we decided to look at designing a clinical trial of home monitoring of creatinine to try to tackle this problem. So working with patients and members of the public to develop our research is something that we do really regularly in digital ECMT already. We seek out input from lots of different groups. One of these is the volunteers who work with the Christie marketing and engagement team, so work with the hospital with a specific team, and they give their time to help us improve all aspects of care at the Christie. Focus groups through this group of volunteers, they take place regularly throughout the year, both online and in person at the Christie Manchester, at Salford Royal and at Oldham as well. We've also conducted lots of online focus groups with volunteers, including from the Kidney Cancer Support Network. So we completed an online focus group with them via Zoom, and that was to help us improve some of our trial documentation. And we've also sought out opportunities to have one-on-one discussions with patients at the Christie and the clinical research facility. So we discuss a wide range of topics when we engage with patients and members of the public. These range from assessment of blood extraction devices to reviewing sampling instructions, having a look at some of the trial documentation with patients to hopefully improve it, such as patient information sheets, and also looking at broader topics such as how should we measure patient quality of life. So when we started to look at the problem of kidney function for patients with a cancer diagnosis, we decided to design a clinical trial, as I mentioned, of home monitoring of creatinine. So we looked at creatinine as we've seen in previous slides, as a sort of key measure that the NHS uses to monitor kidney function. And during the development of this trial, we conducted in-person focus groups, including one at the Christie in Manchester. And during this group, we asked participants to test out a potential device for use in home monitoring. And this device is the one that we did go on to use in the actual trial that we developed. So I will touch on that again a little bit later. And through our kidney specialist colleague, our nephrologist at the Manchester Royal Infirmary, so a separate hospital in Manchester, we also set up a focus group for renal patients specifically, so patients who are there for kidney treatment. And from this, we learned quite a lot about their awareness of kidney function and their willingness to use their own data. So this gave us a really good insight into where patients with a cancer diagnosis might be at the start of the process of doing this type of monitoring. So before we actually looked at the measurement of AKIs in home amongst patients, when we designed our clinical trial and what benefit we can bring to patients by improving the process, we need to make sure that completing home monitoring is something that patients are actually comfortable with. So to make sure we're able to better understand this, we designed the in-home trial to take place in 2 parts, starting with part A. So the primary objectives of this part were firstly, and most importantly, to evaluate if the process of home monitoring that we designed was acceptable and feasible for patients. And we identified a number of ways to measure this, as you can see on the right-hand side of the table here. These included an end-of-trial interview that we completed with every patient who participated in the trial. And really, we included that in the design as a key measure to learn from the patient's experience of home monitoring. We also wanted to understand whether the NHS AKI algorithm that I mentioned briefly previously would be suitable for patients monitoring at home versus applying that to the results that are received in the hospital. So in part B of the trial, as you can see here, we're hoping to continue to learn about this process with a larger group of patients. So we want to keep learning about the feasibility and acceptability of the process. And obviously, with the more patients we can do that with, the better, and get better quality of data. And also hopefully start to look for some early signals of earlier diagnosis of AKI and potentially changes in managing renal function using the home monitoring. So I thought it might be useful to sort of show what the in-home trial actually looks like for patients in terms of its schedule. So the trial was designed to fit around patients' existing visits to hospital. So we essentially, well, I piggyback on planned visits to the Christie Hospital for patients who are coming for different types of cancer treatment. I'll also complete the different trial activities for in-home at the same time. So we initially, when we first designed the trial, looked to target patients who have a diagnosis of types of head and neck cancers to participate in the in-home trial. And this is because this group of patients are particularly at risk of AKI for several different reasons, including side effects of the treatment that patients have for head and neck cancers, and also difficulties with hydration that they tend to have as well. So because of the location of the disease, patients will have both chemotherapy and targeted radiotherapy in these particular areas, so the face, head, and the neck. So as you can imagine, that causes a lot of issues with being able to eat and drink properly because of the different side effects of treatment from targeting so closely to that area. So all these things combine to make head and neck cancer patients sort of a particularly in-need group when it comes to intensive kidney monitoring. So that's our kind of initial target group for the trial. And we've also since branched out into patients with different types of cancers, so patients with urological cancers and also lung cancers as well, as they also receive treatments that can cause problems with the kidneys that's really established in existing research. So as I've mentioned, the trial split into 2 parts, part A and part B. And as you can see from the study flow charts here, they're not that easy to read, but hopefully you can get a bit of an idea of how they're structured. The way these 2 parts work differs slightly. So in part A of the trial, this was a 4-week process for patients. And when patients came to the Christie Hospital to start their treatment, they would also provide their consent to take part in the in-home trial. I would complete detailed training and education with the patients, more on that in a second. Collect some additional blood and urine samples and get the patients all set up to start monitoring alongside treatment. So the way the monitoring works is we ask patients to complete a reading at home themselves of their creatinine on a Monday, Wednesday and a Friday in part A for a 4-week period whilst they're also having treatment. So we start in home at the same time as treatment starts to get a really good baseline of where the patient's kidney function is prior to them starting treatment, so that we can better detect any changes once treatment begins. Then the patients go off and do the monitoring from home. I check in with them during week 1 of trial participation. I collect additional blood and urine samples again at week 4. and then again at the end of the trial. So the idea with the blood and urine samples is that we want to potentially explore if there are other, what we would call biomarkers, that give us an indication of how patients' kidney function is doing at any point in time during their treatment. So as well as asking the patients to monitor their own creatinine, we use these additional samples to explore other potential measures of kidney function as well. And then, as I've mentioned briefly already, at the end of trial participation, every patient that completes the in-home monitoring does a short telephone interview with us, an end of trial interview, where we go through feedback on lots of different aspects of the process, just to learn as much as possible from the patients that have taken part. Because obviously, if we want to determine how feasible it is, we really need to hear in detail from our patients on that. So the recruitment target for Part A was 12 patients. It was a small sort of initial feasibility stage, we would call it. And we ended up recruiting 14 patients in that part, which is great. So based on the findings from that, which have been really positive, again, more on that later, we've now moved into Part B of the trial. So the monitoring process in terms of home monitoring of creatinine is exactly the same for those patients that do complete it in Part B, but they do it for a longer period. So we've extended into a 6-week period to see if it is acceptable to patients to do this for an additional 2 weeks in comparison to Part A. The major difference with Part B is that we're using randomization, 50-50 randomization for the patients who agree to participate. So we've upped our target in Part B to 60 patients with 30 in each arm. And as I say, the patients are randomly allocated to one of 2 arms. So we have one arm where patients essentially continue on their standard of care treatment journey as they would anyway. And then we collect additional data and samples as part of the in-home trial. And then we have the other group of patients who complete the home monitoring process, as I've started to describe, and do the additional blood sampling at home for 6 weeks on a Monday, a Wednesday, and a Friday. So I thought it'd be really good to demonstrate what the trial kit that I've mentioned actually looks like for patients, and practically what the process of in-home blood sampling and monitoring of creatinine looks like. So as I've mentioned briefly already, for all the patients that are taking part in the monitoring in the trial, we provide a full kit, which includes everything that they'll need to take part in the in-home trial. There's some images there that give you a bit of an idea of some of the equipment that patients need to participate. So we go through the entire trial kit, the entire process from start to finish with the patient, and we usually complete the first creatinine reading together in the hospital as the patient's starting treatment. So this involves a finger prick blood test and the use of a creatinine monitoring device called the Nova StatSensor device that patients then use to get that result. So they submit their creatinine reading to us once they have it, and these readings are then reviewed by a nephrologist based at the MRI. They check the results, and as the kidney expert, they know which results might potentially be of concern, and then they will flag these for the patient's care team at the Christie. And that's obviously a really important safety aspect of our trial, to make sure that any data that we do pick up is being actioned if it's needed to. So as I've mentioned, we do provide lots of bits of information to patients, including written information. So a standard aspect of clinical trials is a patient information sheet. So every patient will receive one of these. But what we've also done for the in-home trial is produced a video for patients. And that's based on feedback that we've had during the focus groups, where patients and members of the public feedback on our research, as I've mentioned, as they've often talked about how difficult it is to navigate the amount of written information that you're given as a patient with a diagnosis of cancer when you're attending hospital visits and talking to the specialists there. So this video basically just gives a simple visual guide on how to complete the process of blood sampling at home for creatinine monitoring. And as we've incorporated this into the trial, we've had some really positive feedback from patients on the video. So I thought it would be helpful to play the video for you now, as I think it's a really good way, as I said, of demonstrating the process. So hopefully I'm going to be able to play the trial video for you now. Great. So hopefully that played okay for everybody and you can see me again now. So as you see from the video, there's a lot of steps involved in doing this monitoring, AKI monitoring at home. And the calibration is obviously an additional step before patients then test their own blood sample to produce that creatinine result. And the calibration is something that was required because the device that we selected for the trial, which we felt was the most appropriate based on the focus groups and the feedback we had from patients during the design stage of the trial, is fairly limited in its capabilities. So, and in terms of its registration as well, it's currently approved under all the different regulations for use in the hospital as a point of care device, but it isn't actually approved for use in the home. So again, for safety reasons, that's why this calibration step is involved in the creatinine monitoring in the home. So when we were designing the trial, we did do a focus group that basically followed this entire process with patients. We asked them to complete the calibration and complete the blood testing as well to produce that creatinine reading. And we got lots of useful feedback from patients on that, including some information coming from patients, as you might expect, that there's a lot to think about during those calibration readings. And there's a lot of information that you might need to keep in mind as you're doing those two separate tests before then testing your blood sample. So in the video, you might have noticed that there was a little sort of clear piece of plastic that the person in the video was using to put that calibration liquid onto before they completed that initial test. Based on the feedback from patients on how this could be quite overwhelming or maybe confusing, we developed a little laminated card to replace that clear piece of plastic, which includes all the information that you need to calibrate the device. So it has all the information on what each reading should look like, what you should do if there is an out of range reading, and just acts as a little reminder really, as well as a tool to actually do the calibration testing. And we've had some really great feedback from patients in the end of trial interviews on that. So I think that's a really nice example of how we sort of listen to patients in the design stage of the trial, develop things based on their feedback, and then that was sort of confirmed as being a really positive change by patients that actually took part in the trial as well. So again, as you'll have seen in the video, we developed a smartphone app for the in-home trial to enable patients to submit their creatinine results to us. And this is because, as I've mentioned as well, the trial device can't transmit the creatinine results directly, as it is quite a limited device in terms of its functionality. So you can see in the images here some of the examples of the different steps involved in the use of the trial app. And one of the really key aspects of patient feedback was ease of use of the trial app. So we essentially used part A of the trial to also gather lots of feedback to hopefully improve the app and the design of the app and its use as well. So in the end of trial interviews, we sort of asked detailed questions of the patient, of their experience of the app, including if there are any technical difficulties and ease of use, of course, as well. And also, importantly, any improvements that the patients might be able to suggest that we can make to the app. So based on the feedback of the patients that took part in part A of the in-home trial, we made some direct changes to the functionality of the app that I will mention in my patient feedback in a moment. So I think it's also important to note as well that although for part A of the in-home trial, the sort of key main objective was to make sure that patients accepted the process and that it was feasible, we did also identify 11 different instances of acute kidney injury or AKIs in 6 out of the 14 patients that participated in this stage of the trial. So as you can see here, these were split across 2 different stages of AKI. We did also as well start to see some very early signals that there can be improvements made to patient care based on the data that we collect in the in-home trial. So one nice example that springs to mind for me is the patient that was attending the Christie Hospital for treatment for their cancer on Christmas Eve. So they were attending hospital anyway for a scheduled radiotherapy appointment. And the patient also happened, it was a Monday, and the patient had also happened to have submitted a creatinine reading as part of the in-home trial on that same day, earlier on in the morning. So we picked up on that data. We could see that it was flagging as an AKI. So we sent that information straight across to the patient's care team at the Christie. And whilst the patient was attending hospital anyway for their treatment, the decision was also made by that team to give them some additional fluids to help with their kidney function and help with their overall kidney health whilst they were already there. So although we can't obviously say with certainty that that meant a positive outcome for the patient in terms of their overall health and their ability to stay on treatment, it was just really efficient and a really nice, neat way of us to be able to show that hopefully the data that we collect as part of home monitoring can directly influence the care that the patient receives at the hospital. I'm hoping my next slide is displaying there as well. So as I've mentioned, we get lots of feedback from patients, not just in the design and the initial stages of our trials, but actively during the trial participation process as well, and whilst we're recruiting patients. So patient feedback to date, which has been overwhelmingly positive, which is really great. And it's also provided us with some really key feedback that I've started to touch on already that has changed the actual process of the trial. So in terms of the positive feedback that we got from patients as part of the end of trial interviews, one key bit of positive feedback was the simplicity of the process. Patients did find it easy to do, and they were happy with all aspects of the sampling. The frequency and timing of the test is something that we were really interested to hear back from patients on as well. As I've mentioned, patients did 3 readings on a Monday, Wednesday, and a Friday for the 4 or 6 weeks of their participation. And we found so far that this isn't sort of too frequent for patients. They haven't found it too arduous to do these readings at these time points. The device as well for the trial was generally well accepted, which is obviously really important in terms of getting that creatinine reading. We needed a device that was easy enough to use for patients, and we've found so far that it is, which is great. There were also loads of really helpful suggestions from patients on potential improvements to the trial as well. So some additional details were suggested by patients that took part in Part A of the in-home trial in terms of patient education. So they suggested that we give a little bit more information on creatinine itself in terms of fluctuations in readings, how that it's natural for variations to happen according to time of day, and that the standard ranges of creatinine for female patients is different than male patients. So once this has kind of been discussed as part of the end of trial interview, patients felt that it would be helpful for me to include a bit of extra detail to future patients in that initial education and training session. So that's something I have incorporated since. Another interesting thing that we found is the importance of help of partners and family members. So lots of patients have talked about how it's become a sort of project for themselves and their family to get involved in at home, away from the hospital. And I think actually in terms of the pandemic, as we were recruiting during the early stages of that, it is something that patients and their families could get involved in when perhaps they couldn't come along to help support patients at the hospital. So that was a nice sort of positive thing that we found as part of the trial. Patients also suggested some tweaks to the app, which we did actually make going from Part A into Part B of the trial. So having a copy of their data was one of these, a really good example of these. So patients felt that it'd be nice to be able to see what their previous readings were as part of the trial, to be able to have a look at the changes themselves and to see what fluctuations there might be, perhaps what their normal or average reading might be, as a bit of reassurance for them. So for Part B of the trial, we've actually built functionality into the app where patients can not just submit a new reading, but also look back at previous readings of creatinine as well. So I just thought it would be good to add in as well that my experience of working on the in-home trial as a research practitioner has challenged a lot of my expectations. One of those being that age isn't necessarily an indication of attitudes towards technology. So in Part A of the trial, we had patients from in ages from their 30s through to their 70s. And I didn't really see, although it was just a small group, I didn't really see significant changes in terms of attitudes towards engaging with the app and engaging with technology in general as part of their healthcare according to age. The process of in-home monitoring is something that is seen as straightforward, despite the number of steps involved. So I did have concerns around the number of steps with the calibration tests that patients are required to do, as well as then submitting their own readings. But we found with the right education and training, that is something that patients find to be simple. And another sort of interesting aspect of the feedback and what I've learned from the trial so far for me is that despite these, for some patients, extremely intensive treatment schedules and lots of visits to hospital, there is still this need for additional monitoring to capture these more frequent changes in kidney function. So although patients are at the hospital a lot and they are monitored closely, there are lots of gaps within that where we can potentially pick up on some really key data on their kidney function. So just before I close, I wanted to mention as well what we're looking to do moving forward with our research in digital ECMT. What we're hoping to do up until 2025 as a key aspect of our strategy is to work towards encouraging inclusivity across all of our technology clinical trials and the rest of our work. So although, as I've mentioned, we do quite a lot of patient and public involvement and engagement as part of all of our research, we are aware that we draw from a limited population of participants for those different focus groups and other aspects of involvement who aren't really representative of the Greater Manchester region that we're based in. So as I've touched on earlier as well, there is also a key problem of the digital divide, and this presents an additional barrier to accessing our research with its focus on technology. So it's clear that if we intend for our patients to become our co-researchers, there's lots of work to be done to ensure that this means all patients. So the way we sought to address this was to work with a specialist patient and public involvement organisation in Manchester called Vocal to arrange to design a project that involved a number of different workshops. So in these workshops, we brought together patients, members of the public, members of the team from Vocal and Digital ECMT, academic researchers who specialise in digital health, the chair of the ethnic diversity group at the Christie, and patients and patient representatives, most importantly, as well. And we all came together in a series of workshop discussions, as I mentioned, to look at how we conduct our research from beginning to end and how we might be able to improve and encourage inclusivity and diversity in that. So, as you can see from the diagram here, we structured our workshops around the entire process of a technology clinical trial, and we split this into what we thought were the 10 key steps. And we used this as a starting point to make sure we explored every opportunity possible to improve inclusivity and diversity at every point in the research process. So as the workshops progressed, we were able to identify immediate practical steps to incorporate into our work. Participants gave us lots of very key, quick wins that we could make in terms of encouraging inclusivity and diversity. And beyond these immediate practical steps, we've also looked at the findings of our project, which have enabled us to produce overarching principles, which we can use to guide our team to consider inclusivity in every aspect of our work. And it's our hope that moving forward, the findings from this project will not only improve inclusivity across all of our technology clinical trials in the Greater Manchester region, but will also contribute to the wider discussion of both the digital divide and the lack of representation in clinical research. And that's it from me. Apologies for you all having to listen to me talk for so long, but thank you very much for your time. And if you've got any questions, I'd love to hear them.
Brilliant. Thank you so much, Leanna. Oh, I think we've got a bit of feedback. That was great. I'm absolutely blown away. And I love the way that you've all built listening into the trial itself to actually improve the trials. The fact that something that's that complicated, we all keep talking about having the simplest, the easiest, the most intuitive way of doing things. And yet something that complicated with the right way of presenting it and the right way of educating people could be seen as simple. I think that's quite heartwarming for everyone in this area. And then the extra part about right from the start, even though it wasn't what you were looking for, having the extra data points from home started to make a difference immediately, even in the first few patients. That's something that people talk about again and again. So to see it happening and see it happening naturally, even when it wasn't really the primary thing you were looking for, is amazing. And for each person involved, this is their life. This is their story. So being able to make that a little bit easier for them, a little bit smoother, it's amazing. Thank you. Thank you for sharing that. If it's all right by you, should we go on to questions? We have one here about pictures of other devices that were shown. Has the team at all explored home collections with either Tasso or TAP2?
Yes, we have. So we did conduct another focus group. I think it was once In Home was about to open, where we did look at the HemoLink device from Tasso. So we have evaluated that in particular. I'm not sure about the other device, but we do always keep on the lookout for new devices or other devices out there that we might not be aware of. And we take every opportunity we can to test those with patients as much as possible.
Thank you. And from that, I'm assuming you'll be having feedback on those devices. So if it's somebody that looks after that device, is it okay if they just come and speak to you?
Yeah, absolutely.
Wonderful, wonderful. Okay, the next question, if we move to Just a fundamental question behind the trials themselves. You described at the start how cancer patients don't have the same options and they're not usually allowed in trials. Why?
That's a really good question. So we have something called eligibility criteria that has to be set up for every clinical trial. I'm sure that's true across, every different discipline and not just oncology or cancer treatments. But as far as I can tell, the eligibility criteria, which includes, what a patient's kidney function can be, what all sorts of other major organ function can be, is just based on really, established, perhaps quite out of date data. So it's all just based on previous research that's been done into, what patients, what sort of ranges of organ function patients can have and tolerate different treatments for cancer. So it absolutely needs relooking at in the age of sort of home monitoring and patients using digital technology to collect data on their own health. It's based on old and I would say outdated existing research.
Wonderful. So do you think if this monitoring approach is proven to be useful, do you think that has any potential for actually going back and changing those eligibility criteria in future?
Well, that would be the very, very grand goal of what we're doing, but would be at a point much further in the future when we've done much more research. Unfortunately, these things do take a lot of time. But that was one of the issues that did spark some of the initial conversations around what we can do in home monitoring of kidney function was the really limited eligibility criteria for trials. So now that we have so many technologies that we can use, it would be really amazing if we could make that criteria much more flexible and hopefully allow many more people to take part.
Yeah, I mean, home trials and involving all of that aspect could actually fundamentally broaden them with the eligibility criteria as well as all of the geographical scope, couldn't it?
Yep.
We have another question here that's asking what level of clinical support was required for participants doing home monitoring?
That's a really great question. So obviously for the in-home trial, I'm the dedicated research practitioner. Our technology clinical trials are what I do day in and day out are my focus. So I was always available for support as for any aspect of participation, but not being a registered nurse or a doctor, so a registered clinician myself, there's obviously certain clinical questions that crop up from patients whilst they're on trial that I'm not in a safe position to answer. So I work very closely with the patient's care teams to field any questions they might have in terms of any clinical issues that they're having whilst they're on trial. And I'll sometimes report information directly to the teams as well, rather than just signposting the patient. But in terms of the support the patients require from me, it has been really positive so far in that patients don't need a lot of help from me. I think once we do the initial training and education, as long as we do that right, patients really can go off and be quite independent in this process. But yeah, we do have all sorts of mechanisms at the Christie that I will signpost patients to or find out myself to give them clinical support.
Wonderful, thank you. And we have another question here that's quite a basic question, and this might be from quite a while ago, so you're forgiven if you can't remember. But with the device and the approach linked with the device being so complicated to fresh eyes, what did patients like about it? Why did they pick this one?
Yeah, really good question. I think from the start, unfortunately, we were limited in terms of what creatinine monitors were available at that point and what we could access. To be honest, I think actually the limitations of that device itself do work in its favor for patients. So literally all you can do with this device, it doesn't sort of even have a backlit or color screen. It's very basic. All you can do is complete the calibration test and then complete a creatinine reading. So I think potentially if the device, I mean, the company that makes this device are developing a more advanced version as well. I think the sort of limitations of the device were a positive in the end because there are only so many things you can do with it. So I think, again, as long as that training and education and that support from me is there throughout the participation, it is simple enough to use once you get into it.
So having that anchor is absolutely key. Wonderful. And I think one final question before we close for today. You mentioned diversity in your inclusion work at the end and how you hope that it'll help this trial, but also other trials. Yeah. Will you be releasing that generally so that people in the community will also be able to learn along with you?
Yeah, absolutely. So we've got some short summaries of the project that we'll be sharing, but we're also currently finalising a full report from that project as well that we're going to circulate far and wide as much as possible. So that will be available.
Brilliant. Thank you, Leanna, and thank you for such great questions. Let me move to the next slide, because I'd like to just bring you back to the patient-centric sampling interest group and remind you, if anyone is interested in learning more about the digital ECMT, then please visit the link that Leanna shared in her slides. If anyone is interested in learning more about the patient-centric sampling interest group, or PCSIG, then please visit our website here. The group and the community was created by some amazing people reaching out together across boundaries. And we are all driven in our spare time to embrace this novel and patient-centric way of doing sampling, because it has the potential to reach vulnerable people and extend healthcare to those that need it in a way we just haven't seen before. And the things we're realizing along the way are even beyond what we first thought when we started it. So if you want to join the community, please just search for PCSIG, our acronym, on LinkedIn, and click to follow the company. This is the non-profit one I mentioned earlier. And also ask to join the group. You are welcome. Please join the community, join the conversation. We'd love to have you. The more ideas we have in here, the better. Thank you again to the evidence base. And also, I'm hoping the slide's going to change. There we go. Thank you again to the evidence base and all of our sponsors. And thank you, Leanna, to you and the team for coming along and sharing your research and your story. It is very, very much appreciated. Thank you.
Thank you. Thanks,
Enjoy
Jenny.
your day. Bye.